So we're going to start off the week by going back over enzymes, and we'll talk about enzyme activity. It's a short set of notes, so you'll kind of be writing a little bit more than even what I'm saying because we're going to go over about 4 slides of notes. Then there's a short video for a short response for you to go over, and then there is an essay response. Afterwards, you're going to then do your pre-lab introduction for the photosynthesis lab that you'll watch for the Thursday lesson and write your report on. And then that's all we have for next class. So this week is pretty straightforward, pretty simple, and today is the lighter side. There is a pre-lab quiz though afterwards, and again, it's 21 points worth on the quiz, and it's an open note quiz. So obviously, first things first, the objective is that learners will be able to justify a scientific claim that free energy is required for living systems to maintain organization, grow, or reproduce, and that multiple strategies exist in different living systems. So we're still finishing up enzymes, and we're going to do enzyme activity. As a keynote, since your chapter 6 summary and Learn Smart was due on Monday, I'm going to make your chapter 7 summary and Learn Smart due this coming Sunday. So you'll basically have a majority of the week to complete this. So to define terms in enzyme activity reactions, a cofactor is usually made up of a mineral, and it is a non-protein assistant required by an enzyme in order to function. So examples that I provided are metal ions or coenzymes, and these things have to be attached or have to be present in the environment for the enzyme to work. So you can have the enzyme and you can have the substrate, but some of them require this starting material to combine. So it's almost like the enzyme is a two-part piece or machinery that has to be in contact with each other in order to activate and actually do its job and work on the substrate. So that's a cofactor. Now, getting more specific, a coenzyme is an example of a cofactor, right? But it's specifically a vitamin. And again, it is a non-protein organic molecule, right? Minerals aren't always, minerals aren't organic. Minerals are things like metal ions, things that you would think like when you think rocks, right? Vitamins are organic. So that means they have carbon or hydrogen or oxygen inside of their chemical component, but they're still not a protein. So they can be in the category of a carbohydrate or they can be in the category of a lipid. And they are a molecule that aids in the action of the enzyme. And again, it's when it's bound to the enzyme. So in the picture, you can see that there is a spot here called the active site on the enzyme. And inside of that spot, there's a thing that's already added to it called the cofactor. And here it's acting as an inner lining to the active site. So the cofactor is attached to the active site in this enzyme, and it coats the inside of the opening of the active site. And only when it's coated will the enzyme accept the substrate and work on the substrate. So if you had to have this blue enzyme without the red cofactor attached to the inside of the lining, maybe the shape wouldn't fit with the substrate, or maybe the connections that would be made between the substrate and the enzyme wouldn't match up properly. Or maybe it's just a chemical barrier that doesn't exist properly between the enzyme and the substrate unless the cofactor is present. So there are factors that affect enzymatic speed or how fast enzymes work. Basically, again, we know that enzymes are working in reactions. They're either breaking things apart or they're building things together. And this can happen at multiple different speeds as caused by multiple different factors, right? We've already talked about temperature and pH. And honestly, the only way that we really paid attention to it in our lab was whether or not it works, right? So whether or not the enzyme is permanently denatured, whether it's just been shape altered, or it's just working faster or slower, because, you know, higher temperatures, molecules are bouncing around faster and they're more likely to come in contact. And then at slower temperatures, molecules are moving slowly, so they're less likely to come in contact as often. So we've already observed those. However, there are other factors, including cells regulating the presence or absence of an enzyme. So cells can choose to make enzymes, right? They are proteins, so they are molecules that are organic and they are makeable by cells. So cells will either make more of an enzyme when they need to, or make less of an enzyme when they need to. And when you make more, obviously in the environment, it just means that you have a higher chance of encountering an enzyme by random chance when you're bouncing around inside of a cell. And if they make less, that means that you're less likely, if you're just a particle in the cell or if you're a substrate in the cell, to bounce randomly and come in contact with an enzyme. So that's a cell regulation thing that can affect enzyme speed, or basically what we're talking about is reaction speed, right? Cells can regulate the concentration of an enzyme. So again, how much output they make of an enzyme affects how fast a reaction will happen that is impacted by the enzyme. So the enzyme, if there's a lot of them, again, there's more likelihood that the substrate will encounter the enzyme. And then cells also have the ability to activate or deactivate some enzymes. So like I just said in the previous slide, we talked about cofactors, right? And cofactors basically can be like on and off switches, right? Enzymes sometimes just don't work when there's no cofactor in the environment. And so the cells can already have just a set amount of enzymes inside of its, inside of the cell, right? They've already produced it. They're always around. They never get taken away and put away, and they never get made more. They're just either active or inactive. So the cell can choose to make or intake cofactors. And that's a pretty cool idea, because that way you don't have to keep wasting energy getting rid of and making more enzymes. Instead, you can just, intake this mineral from the environment, or make this vitamin that attaches and activates them. And in case of not wanting to do a specific reaction, you just get rid of those minerals or get rid of those vitamins. And so you don't have to build these complex molecules called enzymes every single time, right? So again, enzyme cofactors, as you can see on the screen, are molecules required to activate enzymes, right? Co-enzymes, again, are non-protein organic molecules. And then vitamins are small organic molecules required in the diet for the synthesis of coenzymes. So that's why we always emphasize that you need to eat your vitamins and minerals to make sure that you're staying healthy. And a lot of those are, we call carbs, lipids, proteins, and amino acids, we call those macronutrients. So you eat a lot of those on a daily basis, usually just by consuming what you normally eat. But we always want like focusing on a healthy diet. We always talk about like eating dark greens, like kale, like spinach, because a lot of those contain vitamins and minerals that you might not be getting. Right? And those are micronutrients. Those are the important ones that, you can have the enzyme that you made because you ingested a lot of protein. But if that enzyme doesn't work because you don't have your vitamins and minerals, then there's no point in you having them in the first place, right? You've made them in your cells, but they're still useless. So that's why we're super focused on having a healthy diet and including a lot of vitamins and minerals in what we consume, because there's no other way for humans for us to get them. So I'm going to show you a video, and basically it's going to be about negative feedback inhibition. And so basically, I'll explain the name of this video, the negative feedback inhibition, after we watch the video, because it literally is what it is. And there's a diagram over here that's also illustrating it, but I'll show the video first. And then you're going to write a few sentences explaining how the production of the amino acid isoleucine produced is regulated by negative feedback inhibition, and that's shown here. So let me move over to this screen. And I need to share the sound for this video so you can actually hear it. And it's probably going to be a little bit loud, so it's up to you if you want to change the volume or not.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
Okay, so basically in this, hold on, stop sharing computer sound. So basically in this animation, they showed you that, yeah, enzymes work in a pathway sometimes, and it's not always just one enzyme producing something and then having that whole reaction end, right? So you'll have a reaction here with the first enzyme when its shape is still conformed to the substrate, and then that substrate, once it changes form, can become the substrate of another enzyme, and so on and so forth. And then there is an end product made. And this end product is important for the cell, right? You're only starting this pathway because it's needed inside of the cell. Now, are you going to infinitely make them forever and ever and ever? No, because usually you're making them for a purpose inside of your cell. And once you have enough of them, you have enough. And you don't want to keep making more because it's wasting resources, wasting time, and wasting energy. So once you've made enough, just by random chance, since the concentration of these products is so high now in the environment, they're going to be randomly bouncing around, and they might encounter that first step, that first enzyme inside of this chain. And like they said, it's an allosteric site. So it's not actually the substrate reaction site. It's not going to be actively competing with the same area that the substrate is going into. And so on the allosteric site, there's a non-competitive inhibitor that's going to come. And that inhibitor, or that thing that stops the reaction, is actually the product of this whole reaction chain. And I feel like this is a really smart way to make a inhibition reaction, because that way you don't have to like wait for another separate product to come. It's actually just the product that you made. And once you've made enough, then because the concentration is high, they're going to have a higher chance of bouncing around and meeting this allosteric site. Now, when it meets the allosteric site, the reason why this is called a negative inhibition pathway is because it's causing a stop. If it was a positive one, it would be causing a go, right? But this is a stop, so it's a negative feedback inhibition. And again, this is on the allosteric site, so it is a non-competitive inhibitor. If it actually came and fit right into the substrate site right up here, then it would be a competitive inhibitor, right? If you fight for the same site as a substrate, then you're competing with them and you're a competitive inhibitor. But in this case, you see the product is a non-competitive inhibitor, so it's coming to this allosteric site on the side. And once it does that, it causes a conformational or a shape change to the enzyme. And as you can see, now the substrate no longer fits in and the reaction will no longer occur. And again, this is a very smart and efficient way to use your resources and not having to wait on another reaction to produce something. So we have the same idea here in this picture where we're looking at the production of isoleucine. And remember, I said negative because it's causing a stop, and it's feedback because it's a loop, right? It's a feedback loop. And it's inhibition because, again, it's causing a stop. Okay, so over here you have the initial substrate, which is threonine, and you have the enzyme, and the threonine fits into its active site just fine. And it's going to go make a bunch of intermediates because, like we said, it's a chain with a bunch of other different enzymes producing another substrate for the next enzyme, producing a substrate for the next enzyme. And once you get to the end, you get your end product, isoleucine. But if you have a lot of isoleucine, then it's going to bind back to this original enzyme over here. And it's actually going to inhibit the production of more isoleucine because now the shape up here at the active site, not the allosteric site, at the active site no longer fits the threonine. So if you can't fit the threonine in there, then the enzyme can't actually react with it anymore and produce this chain that actually makes more isoleucine. So I want you to kind of explain this in your own words now. You can definitely use a diagram to help you, but obviously if the isoleucine is used up or freed up from the enzyme, then that means that there's a need for it again. And that means that since it got used up, there's a low concentration in the environment because even the one attached to the enzyme is being pulled off now. And so now the active site is changing because nothing's attached to this allosteric site anymore. And therefore threonine can reattach to the active site and create that enzyme substrate complex and then go through and have this chain flow again and produce more isoleucine. So the next part here is again the inhibitors that we've been talking about. So there are non-competitive inhibitors and there are competitive inhibitors. So non-competitive inhibitors, like I said, here is the enzyme and here is the substrate. Non-competitive inhibitors have a separate location that they access the enzyme by, and that's called the allosteric site. So the non-competitive inhibitor will touch this allosteric site and cause the active site to change shape just because it's a chemical reaction with the enzyme. And once the active site changes shape, the substrate no longer fits in. And that's just how the reaction is prevented. Now there can be an inhibitor that is the same shape, partially at least, as the substrate, and it can fit into the active site, right? Some enzymes don't have an allosteric site. So sometimes the inhibitor is going to block the active site and actively fight and compete just by random chance of bouncing into the enzyme and block off the active site. And obviously, if there's something there in the active site, then nothing else can approach it and attach to the active site. And that's another way of blocking a reaction from happening or inhibiting a reaction. Okay, most enzyme reactions that are being inhibited are a reversible process because if you permanently change the enzyme, then that would be denaturing it, right? So sometimes when the inhibitor binds, it stays on for a long time, and sometimes it doesn't stay on for a long time. It's all dependent on the reaction that it is related to, how often that product is needed, and the concentration of that product in the environment. If it's low, then the cells are going to want you to make more, you know. So then this enzyme and this inhibitor are going to detach, and then the process that the enzyme originally does is going to continue because the cell needs it. right? So again, competitive inhibition is when the substrate and the inhibitor are both able to bind to the active site. And then the non-competitive inhibitor is when the inhibitor does not bind to the active site, but at an allosteric site, which is on another location. Right, most of the pictures I've shown so far, they're on the active side, on the opposite side of the active site, and that can be true, and it can also just be on another part of the enzyme at any part of the enzyme. It could be close to the active site. They're different shapes to begin with, right? This diamond shape versus this rounded shape, so it's not like they're going to accidentally interact and all accidentally touch the active site and cause that to become a competitive inhibitor, right? The non-competitive inhibitor is always a non-competitive binding site, right, at the allosteric site. It's not going to cause the reaction to stop just because it touches the active site, because it doesn't fit there. So that's pretty cool for specificity. It's like how we're related to what we talked about with the enzymes. Amylase only works with starch and it doesn't work with oil, right, vegetable oil, because the molecules aren't compatible. They just don't fit together. Same idea. So you have your catechol FRQ in a post right under here, and you're going to answer the question. There are multiple parts. You can use your tools in Google Docs, or you can use like your PDF tools to draw into the graph and make sure you label the title and the axes. If you need to, I think a good idea is always to outline what the questions are asking you. So rephrase the questions for yourself, and then outline how you're going to answer each of these questions and break them down, because there are multiple parts of this FRQ. I'll have this graded for you as soon as possible. And then I want you to do your pre-lab quiz for your photosynthesis lab that you'll be doing on Thursday with me as doing the demo. And then the only reason I have a pre-lab quiz for this one too, especially, is because you need to know what the steps that I'm doing in the demo are and why I'm doing them. So it helps to make you kind of, go through them and then rewrite them for yourself in your own words and simplify them so that you understand them better. So that's all I have. Hopefully you can finish this up and get everything done so I can also get it graded for you.